everyone to our HLI seminar series. Thank you so much for joining. For those of you who don't know me, my name is Pamela Brar, and I am the medical director of HLI San Diego. I've been very fortunate to be part of our company since the day we opened our headquarters in San Diego back in 2018. And I will be the host of our seminar today. I want to thank all of our guests on Zoom today for attending, and I hope this is both informative and entertaining. Knowing Rob, it absolutely will be both. And so for some quick housekeeping, the webinar will be recorded today. It's already being recorded. And details will follow via email in the next few days about how to access the recording. So please stay tuned in your email inbox for that. Also, after we've gone through some of our discussion, you'll have the opportunity to ask questions, and you can enter those into your Q&A box there, and we will take some questions time permitting. So we look forward to the questions always. And so with some of that taken care of, it's my pleasure to welcome my longtime friend, Dr. Robert Darling, to our seminar series. Rob, we've been friends for what's over 10 years now, right?
I think so.
Yes. So we knew each other back in the day of when we both had our private concierge medicine practices. And it's so wonderful to get to see you again. And we really appreciate your willingness to share the unique story about your family and genetics and how hopefully it's had a positive impact. For our guests, Dr. Darling has a very unique perspective as a physician himself. And as you will soon hear, as an individual whose family has been personally impacted by the power of genomics. But before we hear about his incredible story, I want you to know him as I know him, who is an esteemed colleague who was the White House doctor for several US presidents, and now has continued to run and develop an incredible, highly esteemed international concierge medicine practice. Time permitting, we'll get to hear some fascinating stories towards the end about his experiences as the White House doctor, which I never can get enough of these stories. So, and then also I wanted to introduce our colleague, Xiaoli Du, who is a PhD geneticist and the director of our medical genomics program. And we may also be joined by our chief of genetics, depending on if he's able to access this from Greece, where it is currently 2 a.m. So, so with all that being said, I will get quiet, and Rob, would love for you to kindly share with us a little bit about how your experience at HLI began.
Absolutely, Pam. Thank you very much for that intro.
Sure.
First, let me just take a few minutes. First, thank you all for joining. It looks like we've got upwards of 60 people on this. I should tell you a little bit about my background because it's kind of relevant to how all of this happened. I grew up on Long Island in New York, did undergraduate there, joined the military to go to medical school in 1981. So I attended medical school at the Uniformed Services University in Bethesda on the grounds of the Bethesda Naval Hospital right across the street from the NIH. Graduated in 85, interned Bethesda, then became a flight surgeon taking care of Navy pilots on aircraft carriers. Got all kinds of neat flight time, and then did some time at the Naval Academy as a doctor, not as a student. And then I completed a residency in emergency medicine in 1994. Took care of Marines for a few years, and then I was asked to consider going to the White House in 1996. Turns out I was the first emergency physician. I went through a kind of a long selection process. Great stories there about how I got picked. But anyway, I was chosen among my peers. I was the first ER doctor to go to the White House in 1996. Spent 3 years there. You know, when I joined the Navy, they said I was going to see history. And I'll tell you what, Bill Clinton provided all sorts of great stories that maybe if we have time to get into.
Oh, we got to make time for that. That's the good stuff.
Yeah, there's some good ones. At that time, I got interested. We were worried about asymmetric threats to the president, to include biological weapons. I started working with the Army at Fort Detrick up in Frederick, Maryland, about 50 miles north of DC. And So when I left, when it was time for me to leave the White House, I was able to convince the Navy to let me go work for the Army at Fort Detrick, because I spent 5 years there study. I was there during anthrax, so that was an eye opener. And then 2 years, I spent my last 2 years in the Navy at the Surgeon General's office, the Navy Surgeon General's office. And then it was 2006 and I retired. So I was 25 years active duty, ran a disaster medicine group out of for my medical school, Uniformed Services, did disaster medicine for several years, and then finally cut the federal umbilical cord after 28 years in government, and started my own private concierge practice. And anyway, during that process, I had a number of patients who were very interested, who became interested in wellness and longevity. And in fact, one knew about HLI and the health nucleus and said,
Wow.
Rob, I want to do that, and I want you to go with me. So that was how many years ago? So that was like 5 or 6 years ago. And so
I think that's right.
Yeah, I went through the health nucleus, and everything was great. I'm in reasonably good health, and with one exception, I came up positive homozygous for the rapsin gene, and which was really kind of interesting because it's associated with something called CMS, congenital myasthenic syndrome. And I'm hoping that when the time is right, both Dr. Dew and Dr. Straticus will jump in with some of the details,
Absolutely.
the genomics of that. So I really was asymptomatic, but here's where the fascinating part comes in, is that my brother, 4 years younger than me, he at the age of 30, he is a construction worker in New York City, doing really kind of hard physical labor, and he starts developing these sort of vague, very subtle weakness. Climbing up a ladder, pulling heavy duty cable. And he was always a kind of a very, muscular, strapping guy. Yeah. And he just thought, he's getting a little out of shape. He actually ignored, so that glided along for 18 months, 2 years, until finally, goes to see his general doc, and they're like scratching their heads, what's going on here? He finally, his symptoms are increasing and worsening, and he ends up. And
he's how old, Rob? How old is he at this point?
Right now, he is 60. He's 60. I'm 64. He's 4 years my junior.
And this, sorry to interrupt you, but this was how he was about what age? You said in his 20s?
He was 30 when he first started developing his symptoms. So that's what's also so bizarre about this, his age of onset. And he actually wasn't. Anyway, so I get my genome sequenced, and so I've got these 2 copies of the rapsin gene, but yet I'm asymptomatic.
Yep, and I'm just showing your genetics report here. Hopefully people can see this on the screen, but this is right. This is your genomics report from from your testing, Robert Darling, up here, and here it is. The rapsin gene, and this homozygous, as you know, means 2 copies of the gene, and this is the disease associated with it. So that I remember was quite a moment.
Well, and so my brother finally, we get him into the top neurologists in New York City at Sloan Kettering, at Cornell. In fact, literally, we're old enough, if anybody's known the book Stupor in Coma, which is a classic in neurology by Dr. Posner. Posner, I forget the other guy, but anyway, he ends up seeing Dr. Posner, who works him up, every muscle biopsies, everything. And he says, after this really detailed workup and referral to other neurologists in New York City, well, we think you are myasthenia gravis, you're seronegative myasthenia gravis. remember, this is like 1990, this is, 2000, 1998, around there. And,
Wow.
but he said, there's something different about you that we don't understand. You're seronegative. And so the treatment was they do a thymectomy. So a thymectomy, crack his chest,
Wow.
right? Take his thymus gland, nothing. Yeah. You post-op a year, they're expecting within a month or so, he's gonna, his weakness, diplopia, things like that he was having would maybe resolve.
Right.
And they got him on cyclosporin. They're putting him, he was treated with super high dose of prednisone. Some days when he would have a worsening of his symptoms, he would be on 100 milligrams of prednisone a day,
Ugh.
alternating with 80. He'd go like that for weeks and months. He would have IV immunoglobulin when things got really bad, plasmapheresis. And I mean, this went on.
Oh my gosh.
So he finally he had to go on disability. He never got better. Pyridostigmine seemed to help a little. He was on pyridostigmine as well. That seemed to actually, but he would have exacerbations of his weakness, and he would have difficulty swallowing sometimes. Occasionally, when he had a real bad crisis, he would have respiratory. He never had to be intubated, fortunately. But this went on for, from age 34, when he was diagnosed as seronegative myasthenia gravis, until when I came to the nucleus, and then we're like, oh my God, when I'm talking to the genetic counselor, well, this is kind of weird. You're like the oldest, they think I'm the oldest person that's ever been described with, having homozygous for the rapsin, and not, I mean, I have these vague, when I get really tired, a lazy eye, I get a little, but other than that, I've never had anything like my brother.
Right.
Anyway, when they see this and they bring my brother in, and then we got a sequence view, and sure enough,
That's unbelievable.
Everybody got involved, and I remember when they came in and said, Chris, we don't have to tell you this, but for the past 25, 30 years, you've been treated for the wrong disease. You've been treated, those steroids you've been taking, the thymectomy,
surgery,
the IVIG, all of that. And his medical bills must have been, I mean, he had great insurance, over $1 million, I'm sure, and then his disability.
Right.
So we got him off of all of that, and then finally, and so really pyridostigmine is the only thing, seems to help a little bit. And I got him into a clinical trial at Hopkins for some candidate drugs that they were, this normally presents, usually it's infantile, or we are really, really unusual in like the age of, my brother was pretty much normal until he was age 30, right? He could do it. So we're kind of unique in the medical literature. So, I mean, the good news is we were able to, stop treating him with some, fortunately, he never developed, he didn't get any diabetes from his prednisone. He didn't get osteoporotic. He was on massive doses for so long. And, he was always trying to be positive. Well, how are you doing?
Right.
Well, it's not that bad. It could be better, but he was always struggling. So,
of course. Well, those are such powerful drugs. And if they weren't even the right decision, and I would imagine, Rob, as a doctor, it must have felt really helpless for you to watch him not improving and knowing that something wasn't quite right about his diagnosis. And I know what that, I mean, that's got to be, it's such a difficult feeling. So
yeah,
I just remember getting the chills down my spine when you told me the story, and it was like, wait, by some crazy, like sort of random thing, I did this testing, found out I have 2 copies of this gene, and the light bulb went off. And then all of a sudden it became something that could help your brother. I can't imagine what was that like for you?
Well, yeah, I mean, the big thing when we know, I mean, he thought that he was probably going to die young, and that probably not from the myasthenia gravis directly necessarily, but just from the complications of all the, I mean, being on it's just amazing that he didn't, develop any diabetes or osteoporosis or all the other things, that you did on the high dose steroids. A couple of times I remember when he had his IVIG, he dropped his pressure. I mean, a couple of times they were giving him he so he had several near misses. But I mean, the good news is that since he's gotten off all of that, the peritostigmine, he can actually ride bikes now and all that kind of stuff. And but he does, like, we're in South Carolina. We live in South Carolina now, hot and humid, and this really kills him. He gets his gait. I can always tell when he's having a bad day because his gait is so. He's probably close where might be starting to think about the walker, but at his age right now, he's psychologically not ready for that, and we'll just have to see how it goes. But but
I'm so sorry. I have a question for you. Had he previously had whole genome sequencing, Chris?
No.
So interesting that he had never, that hadn't been offered.
No. Yeah. He's he's basic kind of a meat and potatoes guy, construction worker, and I remember when I told him we need to do this. He was like, well, what's it going to change? And obviously it changed. I remember, he cried like a baby when they came in and said, Chris, you do not have seronegative myasthenia gravis. You have a genetic disease, CMS, and so the real big thing about this, Pam, there's probably I don't know what the percentage is, but there's probably a number, and Dr. Straticus mentioned this in the paper he did for us and the papers he sent us. There's probably a good percentage of quote unquote seronegative myasthenia gravis, right? So autoimmune disease.
Right.
In fact, who haven't had their genome sequenced, who are probably they probably have CMS, some variation. Right. And so and some of these these patients have gone through the same thing my brother Chris has.
Right.
And, who knows? Maybe some of them develop diabetes or broke a femur from osteoporosis from being on prednisone so long, or it really points to the power of that, because like I say, we could have, had we known he had CMS back 25 years ago, he wouldn't have been on. He would just, all right, so peritostigmine seems to help him. It's not a cure. He's never going to be cured, but the health bill that we would have avoided, right?
Absolutely. And so interesting, right? When you look at the timing of all this, our founder, Craig Venter, was hard at work sequencing the human genome, right? And part of the major movement to accomplish that incredible feat. And it was right around the end of the 90s, that all happened. And so the convergence of all this, right? Because when you and I were in med school, the genome hadn't even been sequenced yet. So which kind of makes us seem old now, sadly. But
it's funny you mentioned the end of the night, because I went with President Clinton when Craig, it was I think it was I want to say it was 1999, some big presentation at the NIH. I was President Clinton's doctor for that, and I'm sure Craig will remember that he shook my hand.
Right, I'm sure he will. Right, he doesn't meet anyone.
No, I got to be there for that, and that was we were just amazed. Wow, they've sequenced the entire. Of course, back then, it cost $100 million or something. And
it was $100 million, yeah. And they had been given, what was it, 10 years at the NIH. And I love. Hold on, I love this. I'll see if I can get this picture up on my screen. But it's a picture of. I'll share my screen really quick, so that you can all see exactly what Rob is talking about. Which is, here we have our founder, Craig Venter, shaking up on the podium with Bill Clinton when they announced the Human Genome Project. So it was a very proud moment as a student of science, right? For all of us. So so incredible to see how that actually, that profound contribution to science and to the world impacts people on an individual basis. It's just like, it's the most inspiring, thing.
Pam, do you remember, do you know that, is there a date? I'm looking for a date. I'm just curious, because I kept a really detailed log of when the.
This was
that's the article.
This was the article, yeah, but it was in 1999. I'm pretty sure. I have to find the exact.
Yeah, no, it was definitely no. I went with him. anytime he leaves the White House, he has to have a doctor and a nurse with him. And I was the one that raised my hand for this one when we went to the NIH. So
right, incredible, just so profound. So, you know, you are one of the most amazing examples of, and you're able to articulate, you know, both as a physician and a patient, so to speak, and the brother of someone impacted. It's just incredible, the story, and inspire. These stories are the ones that inspire us every single day. So, have you found that you've started using genome sequencing in your practice?
yeah, I mean, I have a number of patients. So, you know, I call them the kind of patients I take care of. I call them special needs patients. I have a lot of special needs patients, as you can imagine. And yeah, they're very, very interested. As many of them want to live until they're 117 because
Oh,
life is good. And so, you know, I can't make any guarantees, but no, we've had a few patients go through the nucleus, and you know, it's, you know, amazingly sometimes we find some really interesting things. Other times it's hey, you're everything's right. You chose your parents well, but no, yeah, we've been some are very when it's kind of interesting, and I'm sure you see this a lot more than I do.
mm-hmm.
You know, you get patients with a history of, you know, Alzheimer's or something, or some kind of neurodegenerative, and I don't want to know about that, but I want to know about everything else, you know. So
Right. Yeah.
so I mean, I always feel knowledge is power. When I did this, I said, hey, you know, and I told my brother, I said, you want to know everything. You want to know everything because knowledge is power.
Absolutely.
That's what I tell my patients.
We totally believe in that philosophy. And also what's I find so powerful. I'm sorry, whoever just joined, could you please mute just for a second? Because I think it's
it sounds windy.
Is that Constantine?
Yes.
Oh
finally,
my gosh.
You know, thank you. I have joined. You know, I couldn't join by my computer. I went through several things here, but I've joined by phone. So
oh my gosh, perfect
it was wonderful
timing.
Yeah, thank you.
Constantine, can you just briefly introduce yourself to our audience?
Please, yeah.
Absolutely. So I'm Constantine Stratakis. I work with human longevity now for a year or so, and I'm working with this wonderful group of people that trying to decode genetics and help patients like Dr. Darling. I'm a geneticist. I worked at the NIH just across the Naval Hospital for 30 years. And now I work with human longevity on genetic counseling, interpreting results, doing deep dives with our absolutely wonderful Zaoli Du and others, and of course the wonderful team of clinicians we have at HLI.
Fantastic. Well, we can't thank you enough. Is it really 2 a.m.? Are you in Greece?
Yes, it's 2 a.m. It's in Greece. I'm on an island. And, you know, because I was traveling, I didn't have my HLI computer with me. So I had to use my other computer, but apparently the system is impenetrable, which I think it's good, actually.
Oh, that's a good sign. Absolutely. Well, we are so grateful that you're joining us. And Constantine, I know this had to have been a very interesting case for you as a geneticist. I would love to hear your perspective on this experience, you know, and what you learned and how what your reflections are.
Absolutely. I think this is, first of all, Dr. Darling said it all in terms of how a disease, the same mutations can present with two completely different phenotypes here between Dr. Darling and his brother. As Dr. Darling mentioned, these mutations in congenital myasthenic syndrome, in CMS, they present very rarely with a mild phenotype such as a droopy eyelid that Dr. Darling had, very rarely. Most of the time, there are serious symptoms, and I shared some of the publications when we first met with Dr. Darling. 2/3 of the patients appear to present with mild to severe symptoms, and only 1/3 with very severe disease. Very few patients with these types of mutations present with almost nothing, like Dr. Darling had. So it is these patients that often get misdiagnosed later in life with, for example, myasthenia gravis. Even the mild to severe patients get diagnosed with myasthenia gravis. So the lessons here are many. One, the different phenotypes, the same mutations can present even in the same genetic background. I mean, two brothers share 50% of their genetics. And so we see that in the same genetic background, we can see the same mutations presenting with two different phenotypes. So that's lesson one. Lesson two, if you have two diseases presenting with similar phenotypes, if you don't think of the alternatives, you can misdiagnose and mistreat the disease. In this case, CMS was misdiagnosed as myasthenia gravis. And given the treatment for myasthenia gravis, which is not necessarily without any side effects. So we searched the literature and found out that several patients with late onset CMS, congenital myasthenic syndrome, have been diagnosed with antibody-negative myasthenia gravis. And so today, in experience centers, they are aware of this, and they're looking for CMS among their patients that are antibody-negative myasthenia gravis. So that's lesson number two. Lesson number three, and perhaps the most important one, when you have the right diagnosis, you're able to give the right treatment and avoid the side effects of the wrong treatments. And I think that says it all.
That is incredible. Absolutely. So, Konstantin, I'm sure for you, starting out your career in genetics, this was not the way genetics was necessarily applied, I would imagine, when you started your career, right? The notion of screening ostensibly healthy people and learning things that you didn't expect to find. I'm sure it's been a very interesting journey for you as a geneticist as well.
Absolutely. When we all started in genetics, we had very few opportunities, first of all, to treat patients that had a known genetic disease. Most of the patients we knew that had known genetic diseases had severe metabolic syndromes and other neurological conditions that for which there was no treatment. Today, and I'm sorry for the wind here, today, not only were able to offer treatment, targeted treatment, that's the important thing. So not just offering treatment, but offering targeted treatment. So now trying to treat the actual genetic defect in so many diseases, but also, as you mentioned, we're able to do genetics on people that are not necessarily so sick, or patients or people that come to us for prevention. So we are able to do the genetics. We're able to find out what predispositions they have and offer advice to avoid disease, or if the genetic defect is certain to give disease at some point, actually do something that will mitigate or completely avoid the appearance of the disease.
Amazing. Such a powerful science, and to be able to apply it to actual individuals is so exciting. I know for me as an internist, I never thought during my days of practice I would be able to access this type of genetic data and learn how to use it and get comfortable with it. And I think for me, one of the most powerful parts of the knowledge that we acquire at HLI is the fact that not only do we get the genetic reports, right, the whole genome sequence for any individual, but we also are able to collect all of this physical information in a very efficient manner, basically learning about the brain, the heart, the entire body, the organ systems, through imaging, through this whole genome sequencing and blood work. It's such an amazing view into someone's health and into things that might potentially be a problem, or are a problem that are, as this case well describes, that aren't clearly characterized or properly diagnosed. So we're very fortunate, I think, to have the ability to provide this to our patients, and also it's a really fascinating way to practice and something I feel very proud to be a part of. So this is a wonderful example of being able to help your own. Rob, I feel like we're longtime friends, right? So this was, I know for me, very special and gratifying. And to know your brother seems like he's doing well, hanging in there.
Yeah, I mean, I think I would venture to say that had he been on the high dose steroids and the intermittent plasmapheresis, IVIG, cyclosporin, I don't know where he would be today. So it's been 5 years he's been off all of those things. The pyridostigmine seems to be the only, it works. And as Dr. Straticus mentioned, that was kind of diagnostic of the fact that it works for him is more proof that, of the CMS component of his clinical picture. So I don't know where we would be right now if we hadn't done this. And I would love, I'd love to see, I don't know if there's any data out there on all these other seronegative myasthenics out there who, haven't had their genome sequenced. If maybe they're in the same position, there's probably a lot of additional patients that could be helped. I don't know what data exists on it. I don't know if Dr. Dew, Dr. Straticus can comment on that.
Well, as I mentioned, there are a couple of papers that looked at a series of patients that are antibody-negative myasthenia gravis, and a few of them were identified as having mutations, especially in RAPS1, this particular gene. I think, one of the things that could be done is reach out to, because there are myasthenia gravis support groups, and reach out to the support groups and identify the patients that are antibody negative, and then simply inform them about the possibilities that exist. So that's, I think, something that we could certainly do.
Absolutely. Absolutely. And we take questions, on a daily basis from potential clients, existing clients, family members, et cetera, about how we might be able to guide them in the right direction. I do have a couple questions coming in, and Constantine, I would love to hear your thoughts on this. So one question is, how is genetics applied after a person has been diagnosed with cancer? Are various cancers different from a genetic perspective? Would love to hear your thoughts on that.
So the question is, how are various cancers different from a genetic perspective?
That's one of the questions. Yep.
All right. So, we all get cancers, regardless of whether we have a genetic predisposition or not. What is important to remember is that cancer itself is always a genetic disease. In other words, cancer develops from cells in our body, parts of our body that develop genetic defects. So regardless of whether we were born with a genetic defect, the actual cancer cell that starts the cancer always gets a mutation, always gets a genetic defect. So cancer is always a genetic disease. Now, what happens is that some of us have a predisposition to assist in this process. Basically, we have in other words, something in our genome, something in our genetics that helps our cells to take that wrong path, to develop additional genetic defects to go on and develop cancer. And so these types of cancers that develop in people that have these predispositions are inherently different from the cancers that develop in people that don't have the predisposition, because they start off with a very different system of defects. And that is why it is very important to know whether a cancer, and it can be a common cancer, it can be a rare cancer, it can be any type of cancer. That is why it is very important to know whether that cancer, breast cancer, prostate cancer, whatever, has developed in a person that has a genetic predisposition. Most of the time, the types of treatments, the prognosis, how long the patient will be treated for that tumor, what type of therapy will be given, most of the time will be different between the people that have the predisposition and those that don't have the predisposition.
Got it. Right. And so I remember when I first came to HLI 8 years ago, and I was refreshing and learning a lot about genetics, being an internal medicine doctor, more of a generalist, right? The concept of, as you're pointing out, there's the germline, which is the genetics that we're born with, right? What we inherit. and those, there's germline mutations or variations that can happen that predispose us. So those are things like BRCA1, BRCA2 genes, et cetera. And then there are those genetic changes that happen to us as a result of some potentially environmental things. And my understanding is we call those somatic changes to the genetics. So in other words, there's genes that we inherit from our family, and then there are genetic changes that can occur once we already exist. And those are different. It's a different type of genetic issue. Is that fair to say?
Yeah, it's absolutely true. So as I mentioned before, all the time, cancer is genetic. Sometimes the genetics is something we inherited. Most of the time, the genetics is something that happens for a variety of reasons. And environmental factors play a big role in the development of these genetic changes.
Right. And I think so. One of the attendees is saying this is a reference to multiple myeloma. So maybe I think the previous question is referencing multiple myeloma. So multiple myeloma is an example of a blood cancer, obviously, and that affects potentially can lead to a pretty significant systemic type of illness. Any comments, Konstantin, on the genetics related to myeloma?
There are a few predisposition genes now for multiple myeloma, and it's not different from other types of tumors where the majority of the tumors, the majority of cancers are developing without known predispositions due probably, as we discussed, to environmental factors and exposures. And in multiple myeloma too, there's a few genes that have now been identified that predispose the development of multiple myeloma. However, unlike, for example, breast cancer, these predisposition genes are not as strong. So in other words, I'm not aware of a gene that predisposes to multiple myeloma in, for example, 80% of the patients that have the gene defect. That's the case for BRCA1 or BRCA2 in breast cancer. What we know about the types of genes that lead to multiple myeloma is less certain and less potent than other inherited factors for other tumors.
I see. And since I think cancer is such a relevant topic for so many of us, Konstantin, can you briefly comment on when a new cancer is diagnosed? What are some of the new methods that we might have to select targeted treatments? Like, in other words, the notion of sequencing the tumor's genome.
Absolutely. So it's very important today to study the tissue from the tumor that develops. And we can do this with a variety of ways, searching for what we call in medicine biomarkers. So among the various biomarkers we use to characterize a tumor is, of course, the genetics of the tumor. So we can do immunohistochemistry, we can measure proteins that are produced by the tumor. These are all biomarkers. And these are all used for determining the right type of therapy. But perhaps one of the most powerful biomarkers that characterize a tumor is the genetics of the tumor. What we do is we sequence the biopsy, the cells that were received from the biopsy, the same way we sequence the blood cells when we take a blood sample and we identify the DNA of an individual, so exactly the same way. And what we find out is gene variations that are unique to the tumor. Those gene variations that are unique to the tumor, along with the other biomarkers that I mentioned, such as, for example, immunohistochemistry and serum markers, those are all used to determine what we call targeted treatment. And this is today common practice in tumors such as prostate cancer, breast cancer, head and neck cancer, and so forth.
Targeted treatments. Got it. Based on the genetics of the tumor versus what the...
And other biomarkers.
Yes. That's incredible. I think we have time for one more question, and then I want to leave just a few minutes for Dr. Darling to share some of his interesting stories about being a White House doctor. So one more question, also related to cancer. A podcast spoke about how cancer is also a metabolic disease. The doctor said all cancers have damaged mitochondria. What is your opinion?
The mitochondria are very important organelles of the cell that are essential both for normal cells and for cancer cells. They are essential because they are the factories of energy for the cells. So they produce all the energy that the cancer cell needs to grow.
Yes.
So it is true that mitochondria in cancer cells behave differently than mitochondria in normal cells. However, I wouldn't necessarily say that they're damaged. What they are, they use a different system in providing the energy for the cancer cells to grow. So they're not necessarily damaged. Now, there are some tumors where there are damaged mitochondria, but they're rare. Most of the time, cancer cells need mitochondria to grow because they are the factory of energy for the cancer cell, just like they are the factory of energy for the normal cell. But it is absolutely true that mitochondria in cancer cells develop mechanisms that provide this energy through other ways, and those mechanisms can be actually molecularly targeted. And we use that information today to target the mitochondria in cancer cells, or the way these mitochondria work in cancer cells, so that we can attack the tumors.
Excellent. That's very helpful. One last question, and from one of our attendees, any comments on whether diet can affect the progression of cancers?
Well, yeah, for sure. I mean, we know that patients that receive chemotherapy need certain types of diets, and that if they don't, or if they are exposed to a different diet, their rate of progression of the tumor is different. So that's so number one, patients that are already receiving chemotherapy need to get the proper diets that are prescribed by their doctors in order to have successful responses, or more successful responses to chemotherapy. Number two, I mean, in general, diet as an environmental factor is important for the exposures we have. And so diets can influence the development of a tumor. They can also influence, of course, the progression of a tumor. That's particularly true for tumors of the GI tract, like pancreatic cancer, colon cancer. It's not necessarily true for other types of tumors, but especially for tumors of the GI tract, diet is very important.
That is so helpful. Thank you so much, Konstantin. Really appreciate you're taking the time and being such a trooper to join us at such an early hour. We really appreciate you. Love having you as part of our team. Shali, thanks also for being on as well. So with that, I'd like to allow Dr. Darling to share tell us about a couple of your most interesting favorite stories about your time in the White House.
Well, it's interesting that we should be talking about genetics and laboratory testing, because one of my stories at the White House involves exactly that. Recall that I was at the White House 1996 to 1999. Well, what happened during that period that was of most? Well, impeachment.
Wow.
Impeachment seemed to be routine now. We've had all that, but when President Clinton was impeached, it was an amazing time. I was there for that, and I don't know how many news hounds we have here, but
at least one.
Okay, well, I was on duty one day. So when you were on duty, when the president was in the White House, there was a duty doctor. Usually on the ground floor. The doctor's office has been there since 1922. And so it happened to be one day I was the duty doctor. My boss, Dr. Connie Mariano, was
my friend.
You know, Connie, yeah, yeah.
I love Connie.
Connie and I are great friends. And anyway, one day there's a knock on the doctor's on the door, and I go and answer it, and it's David Kendall. If anybody knows, David Kendall was one of the attorneys that defended President Clinton during impeachment, and he's this was pre 911, so it was pretty easy. People walked around. It was get into the White. It wasn't so difficult. Anyway, may I speak to Dr. Mariano? And I go, Connie, David Kendall's here. I bring him in. She closes the door. They're in there for 20, 30 minutes. He leaves. She calls me in, and she says to me, Rob, do you know what PCR is? I said, yeah, that's polymerase chain reaction. That's where you have a little bit of DNA and you can figure out where it came from.
Oh.
So this is like, I don't know, when was this? 98 or something like that. And Kerry Mullis from San Diego got I believe it was 93. He got the Nobel Prize for figuring out how PCR. Yes. And so Connie, anyway, I said, yeah. She says to me, Rob, they have a dress, a blue dress with his essence on it. And so that, if you remember the blue dress, and that's where they kind of figured over it. So remember, I didn't have any NDAs, and what are they going to do? And I think that's been written in several different places. So
yes. Oh my goodness.
So I had a ground floor seat to that, and of course that led to him. I guess he got a subpoena to submit a blood sample. Thankfully, I wasn't asked to draw it, but yeah. So
wow, that's a whole nother world of. They didn't teach us about that in med school or residency.
Yeah. I have a lot of other really colorful ones, but I'm looking at this list and I don't know most of these people, so I'd have to be enticed with some wine or bourbon but there's another. I mean, I've got so many. There's another. Again, these are all Googleable.
Right.
So here's another good one. So again, history. There were two events that occurred on the same day, and I think it was in 1998. There was when Newt Gingrich, who was the Speaker of the House, resigned, and that was the same day. Remember Senator John Glenn, who was an astronaut?
Oh, I love John Glenn. Yes.
He went up into outer space in the space shuttle.
Met him a couple of times. Yeah.
Yeah.
Amazing.
And so I was with the president in Arkansas. He was visiting one of his friends named Bubba. And of course, anytime President of the United States goes anywhere, it's not like the limo and there's like 50 Secret Service armed guys, the doctor, right, the nurse, staff. There's literally 50 people. Anyway, so Newt Gingrich resigns, and then wow, John Glenn wants to send President Clinton an email from outer space. Okay. And we're at this guy's house, and I can remember the chief of staff, John Podesta, telling this comes to Podesta. Podesta goes to President Clinton, and Clinton says, "Make it happen." And they look at WACA, that's the White House Communications Agency. These are all Army guys. Okay, the president can launch a nuclear weapon from anywhere he is at any time of the day, 24/7. But at this particular time, he wanted to send. We had 1 hour because President or Senator Glenn was going to be circling the Earth, and they said, told Walker to get a laptop. Nobody had a laptop except one guy, Dr. Darwin. So I basically I tell NASA I have my 25-pound Toshiba laptop. I tell NASA send it to rdarlingmd@aol.com.
Oh my gosh.
So John Glenn sends this email from outer space to NASA. NASA sends it to rdarlingmd@aol.
Oh my God.
Down to my computer. He reads it, and he sent 2 emails in his 8 years as president. 2 emails. He responds to John Glenn on my laptop. It took about half an hour because he used both index fingers and he wrote like 2 or 3 paragraphs.
Oh, it took a half an hour to actually type it? Okay.
Type it, yeah. And the White House photographer there is taking pictures, gets up there. Next day, they do a press release and with John Glenn's email, his email, And then this is about, I don't know, 3 or 4 months before I'm ready to leave the White House. And to get the president to sign anything was a really big deal. You had to ask about 20 different people to
Oh,
get,
wow.
okay. But we used to give him allergy shots. So, and I used to give it to him. So I just like cut through all the BS. One time, we getting out of the shower, I call the valet. Mr. President, would you mind signing this? Remember that email you sent, John Glenn? Oh yeah, Doc, can I get a copy of that? Anyway, so that's all. You can Google all that. And I ended up selling that laptop because I had 3 kids in private school. I actually on Christie's, because that particular laptop and then the picture and all that kind of stuff he did. I needed. Well, I figured the value was the computer itself because of the electrons and AOL. Right. That thing degrades. So anyway, so that's actually a clean story. But all the other ones, I don't know any of these people, so they're going to have to buy me a drink or something because there's all computers that are amazing.
Well, that is just absolutely priceless. I love that story. And yeah, we're very fortunate to get to practice and meet a lot of fascinating characters. And that is very true I don't have anything quite that juicy, but I'm very lucky to be in the world that we're in and taking care of people that are engaged in their health and curious. And it makes what we do incredibly gratifying. And well, I want to just thank you so much, Rob. I know how busy you are, and Constantine, much gratitude for your time and expertise and for being part of our team. We are here at HLI to take questions, to support our clients and families and anyone that might be interested in learning more about what we do. You'll, if you're on this webinar, you are on our email list. So we'll be sending out a follow-up with a link to the recording in the coming days. We're very grateful that you're all joining us. The questions make our sort of widen our awareness about what people are interested in, and we really appreciate your participation. We'll be having future webinars on a very regular basis, and we'll be trying to send out advance notice because we know how busy our clients are. So thank you for growing with us. We are grateful for you and look forward to seeing you back many times to come. Keep you healthy and well in the future. And Rob?
Pam, just one thing. I don't know if people have access to, if anybody has an individual question, you send me an email or something like that, or I'm happy to have a chat or whatever. So and thank you for asking me to do that. I enjoyed this. I hope everybody else enjoyed it. And maybe when I'm in San Diego, next wherever people are, I can share some of my other stories. And
absolutely, we love that. Let's see, 1 second. I think I have one more. Let's see if I can figure out how to. Let's see, Judith. I'm going to allow you to talk. We have a question from one of our attendees. I think you might be muted. Yeah.
Can she unmute herself, though? That's the question. Oh, there you go.
Not on my cell phone. I just had to unmute. Sorry.
No worries. Thanks so much.
I just wanted to thank you for the program. I really enjoyed it. And while I don't have any of these issues, it's always good to learn. I love learning. So thank you very much.
Thank you so much for that. We really appreciate it. It means a lot to us. And we're so excited to do more of these and to learn from you as well. So thank you.
Okay. Thank you.
Thank you for sharing. We really appreciate it. And everyone, I hope you all have a wonderful night, and we will see you again soon.
Bye-bye. Out
Bye.
here.
Thank you again. Bye-bye.